Okay, let's get into this. Forget the idea that you just need to pump cancer cells full of iron to kill them. What's really surprising from this Nature paper is the idea that the real key might be activating the iron that's already inside their lysosomes.
Exactly.
It's like flipping a switch on an internal self-destruct sequence, triggering this process called ferroptosis.
That's right. And ferroptosis, it's become a really hot topic in cancer research. It's not like typical programmed cell death, apoptosis.
Right.
It's driven by something very specific, this iron-dependent chain reaction that chews up the lipids, the fats in cell membranes, a kind of oxidative damage cascade.
Okay.
And what's been sort of hinted at is that cancer cells actually need their lysosomes, the cell's recycling centers, to be working properly for ferroptosis to even happen. That suggested the iron in them could be crucial.
Right. We sort of knew lysosomes were involved, and
Yeah.
maybe blocking iron from getting out could trigger damage.
Mm.
But the big question was always, where does it start? Where's the spark? So today, we're diving deep into this Nature article. We want to understand how this lysosomal iron gets switched on in cancer cells. And maybe more importantly, can we hijack that switch? Force these cells into ferroptosis?
Yeah, and the researchers followed some really interesting leads. It all starts, perhaps counterintuitively, with a molecule known to protect cells from ferroptosis.
Ah, okay. So leproxtatin 1, or LIP1, that's the protector molecule.
That's the one, a well-known ferroptosis suppressor. And the researchers wanted to figure out how it was protecting the cells. What was its mechanism?
So how did they track it?
Well, they got clever. They made a slightly tweaked version, CLIP1, which had a kind of chemical handle, a click tag, so they could visualize it with fluorescence microscopy.
Like a tiny GPS tracker for the molecule.
Exactly.
Yeah.
And when they put this CLIP1 onto various cancer cells, fibrosarcoma, pancreatic, lung, you name it, they saw something striking.
What was that?
It wasn't just floating around. It was specifically gathering, accumulating right inside the lysosomes, really concentrated there, even at high doses.
Wow, okay. So it zeroed in on the lysosomes. That immediately points to the action happening there, right?
It strongly suggested it. And it wasn't just in cell cultures. They looked in vivo, in mice with acute kidney failure caused by problems with a key ferroptosis regulator protein called GPX4. Giving these mice CLIP1 helped them survive better. And when they looked at the kidney tissue, same story. The CLIP1 was piled up in the lysosomes of the kidney cells.
Okay, and critically, this tagged version, CLIP1, it still worked like the original LIP1. It stopped the lipid damage.
Yes, exactly. It prevented that lipid oxidation and protected cells when GPX4 was knocked out or inhibited with a drug called RSL3.
Yeah.
So CLIP1 was a reliable stand-in, letting them see where LIP1 was working inside the lysosomes.
All right, so LIP1 is in the lysosomes, stopping ferroptosis. But what is it actually doing in there? Is it just mopping up radicals?
Well, that's where things got really interesting. LIP1 didn't quite fit the usual profile. Normally, for these ferroptosis suppressors, how well they directly neutralize damaging free radicals, their radical-trapping antioxidant activity, or RTA, correlates strongly with how well they stop ferroptosis.
But LIP1 was different.
It didn't follow that rule perfectly, which hinted, maybe there's something else going on, something beyond just acting as a simple antioxidant.
And given it hangs out in lysosomes, which are full of stuff, including iron.
Precisely. Lysosomes are acidic and known to contain iron. And LIP1 has nitrogen atoms in its structure, which are known to interact with metals. So they hypothesized maybe LIP1 is directly interacting with the lysosomal iron.
Okay, makes sense. How did they test that?
They used NMR spectroscopy. It's a technique that basically uses powerful magnets to see how molecules are bumping into each other, how they interact at an atomic level.
And what do the NMRs show?
Clear evidence. LIP1 was directly binding to iron. That's the oxidized form of iron. They saw different binding ratios, too, sometimes 1-to-1, sometimes 2 LIP1 molecules grabbing onto one iron ion. And importantly, this binding was much stronger under acidic conditions mimicking the inside of a lysosome. Plus, it was reversible. It could bind and unbind depending on the pH.
So LIP1 grabs onto iron in the acidic lysosome. How does grabbing iron stop the lipid damage, stop ferroptosis?
Good question. They used another technique, cyclic voltammetry, which measures sort of the electrochemical personality of iron, its tendency to gain or lose electrons and react.
Okay.
And they found LIP1 actually changes iron's redox potential. It makes it less chemically reactive in that context.
It calms the iron down, essentially.
That's a good way to put it. And they back this up by looking at related molecules. One called METCLIP1, it still got into lysosomes, maybe even better, and it had a stronger effect on iron's redox state. and it was a better inhibitor of lipid oxidation. Another one, Al-CLIP1, didn't get into lysosomes as well, had a weaker effect on iron, and guess what? It was a worse ferroptosis inhibitor.
That connects the dots nicely. Lysosomal location, iron interaction, and ferroptosis protection all line up.
Exactly. And one more piece of evidence, they tagged defroxamine, DFO, which is a known iron chelator that actually promotes ferroptosis.
Right, the opposite effect.
And that tag DFO also went straight to the lysosomes. It all points to lysosomal iron being the critical factor.
Okay, so LIP1 messes with the iron. But what about the lysosome environment itself? That acidity you mentioned, does that play a role?
Absolutely. If lysosomal iron is key, then changing the lysosome's environment should matter, right? So they use drugs like hydroxychloroquine and bafomycin A1. These drugs raise the lysosomal pH, make it less acidic.
And what happened when they did that?
They saw less chelatable iron and less of the reactive iron too inside the lysosomes. And the consequence, the cells became resistant to lipid oxidation triggered by RSL3.
Wow. So just making the lysosome less acidic protects the cell, likely by changing the iron state.
That's the idea. And LIP1 itself had similar protective effects, which fits perfectly with it interacting with that lysosomal iron. They even used a special fluorescent probe that lights up only when it binds iron. In a test tube, adding LIP1 dimmed the fluorescence when iron was present. More evidence for that direct interaction.
Okay, the case for LIP1 working by engaging lysosomal iron seems really strong. So if the action starts there, can we see the damage starting there too?
That was the next logical question. Where does the lipid peroxidation, the signature damage of ferroptosis, actually begin?
How do they look for that?
They used fluorescent probes again, but this time ones that glow when lipids get oxidized. Liverfluo is one, Botapy 665676 is another. They treated cells with RSL3, that GPX4 inhibitor, and watched where the fluorescence popped up over time.
And what did they see?
It was pretty clear. After a short RSL3 treatment, say just 1 hour, the glow, the lipid oxidation signal, was mostly coming from the lysosomes.
Yeah.
Ah, okay. Predominantly there first.
Yes. But if they waited longer, maybe 4 hours, the signal started spreading to other parts of the cell, like the endoplasmic reticulum.
Which strongly suggests the lysosomes are ground zero. The fire starts there before spreading.
Exactly. And consistent with that, they also found that various ferroptosis inducers caused levels of glutathione, a key cellular antioxidant, to drop specifically in lysosomes, while harmful oxygen radicals increased there. It all converged on the lysosome as the initiation site.
Okay, so they've built this really detailed picture. LIP1 works in the lysosome by interacting with iron. The lysosomal environment affects the iron, and the damage seems to start right there.
Precisely. And having established all that, they took a really bold, innovative step.
Which was?
They decided, okay, if lysosomal iron is the trigger, can we design a molecule to intentionally pull that trigger, to activate that iron and force ferroptosis?
Ah, okay. So instead of inhibiting it, they want to induce it using the same location. This is where Fento-1 comes in.
Exactly. Fento-1. It sounds like something out of science fiction almost. What is it? How's it built?
It's cleverly designed. It has 2 main parts linked together. One part is derived from marmycin, a molecule known to accumulate in lysosomes. That's the targeting part.
Gets it to the right place.
Right. And the other part is a Chen-White ligand, which is known to interact with and activate iron. That's the payload. So deliver an iron activator specifically to the lysosome.
A targeted weapon. Did it work? Did it go to the lysosomes?
It did. The data showed it rapidly localized to lysosomes. And once there, it did exactly what they hoped.
Yeah.
It triggered a big increase in lipid oxidation.
You could see the damage.
Oh, yeah. They saw markers like 4-H&E go up and signs that the membrane phospholipids themselves were being broken down. Things like lysophospholipids and glycerol were produced. And the bottom line, this led to cell death.
But was it definitely ferroptosis, not some other kind of cell death?
Good point. They checked that carefully. The cell death caused by Fento-1 could be blocked by known ferroptosis inhibitors, iron chelators, those radical-trapping antioxidants we talked about. But inhibitors of other pathways like apoptosis or necroptosis, they had no effect. Fento-1-induced death was specific to ferroptosis.
That's convincing.
And even more so, Fento-1 was synthetically lethal with GPX4 inhibitors, meaning if you were already pushing the cell towards ferroptosis by blocking GPX4, adding Fento-1 was devastatingly effective. Further proof it works through the ferroptosis pathway.
Right. And did they try variations?
They did. They made Fento-2, -3, -4, -5, slightly different structures, and they found their potency, how well they killed cells, correlated with how well they could promote proximity between that iron-activating part and the membrane lipids inside the lysosome, and how reactive that iron catalyst part was.
So they really engineered a specific lysosomal ferroptosis inducer, a molecular Trojan horse, almost.
That's a great description.
Okay, that's incredibly cool from a molecular design perspective. But what about cancer? You mentioned aggressive tumors earlier. Is there a link here?
Absolutely. This is where it gets very clinically relevant. They looked at actual primary tumor tissues from patients' pancreatic cancer, sarcoma, metastatic breast cancer.
And what did they find in those tissues?
Compared to nearby healthy tissue, the cancer tissues had significantly higher levels of total iron.
Which kind of makes sense, right? Cancer cells are rapidly dividing. They need more resources, including iron.
Exactly. But they went further. They looked at a cell surface marker called CD44. High CD44 levels are often found on these really nasty cancer cells. They call them mesenchymal, drug-tolerant persister cells, or DTPs.
The ones that survive chemo and spread.
Those are the ones, major drivers of metastasis and treatment failure, and they found a direct correlation. Cells with high CD44 had more total iron, yes, but critically they also had more redox active iron, specifically inside their lysosomes.
So those really tough drug-dodging cells are potentially walking around with lysosomes packed full of this readily activatable iron.
Precisely. It's like their strength-needing iron for their aggressive behavior might also be their hidden weakness, an Achilles heel.
Which sounds like a perfect target for Fento-1.
You got it. When they took cells directly from patient tumors and treated them with Fento-1, what happened? They saw lipid oxidation and cell breakdown, specifically in those CD44 high cells. Fento-1 preferentially killed them off.
Wow.
And Fento-1 was also more potent against DTP-like cells that had survived chemo treatment in vitro. It even worked synergistically, boosting the effect of standard chemo drugs in pancreatic cancer cells.
Did they test it in an animal model? Like in a living system?
Yes, they did. They used a mouse model where breast cancer had spread to the lymph nodes, a common site for metastasis.
Yeah.
They injected Fento-1 directly into those affected lymph nodes, and they saw reduced tumor growth. And again, the effects seemed targeted. They saw lipid oxidation happening preferentially in the CD44 high cancer cells within those lymph nodes.
That's pretty strong in vivo evidence supporting the whole concept.
It really is. It ties it all together.
So the picture emerging is CD44 high cells take up more iron, load up their lysosomes.
Yeah.
They use this iron, sure, maybe for growth, for resisting drugs, but it creates this vulnerability. This pool of lysosomal iron is just sitting there, ready to be activated by something like Fento-1, triggering self-destruction via ferroptosis.
That sums it up perfectly. It's exploiting a metabolic adaptation of these aggressive cells. Although they did note something important.
What's that?
Using very low sublethal doses of Fento-1 could actually make cells resistant to ferroptosis down the line, which suggests that just hitting them once might not be enough. Combination therapies might be needed to really wipe them out and prevent resistance.
That makes sense. Cancer cells are adaptable. So thinking about the impact of this work, it really feels like it's answered a big question about ferroptosis, hasn't it, where it kicks off?
I think so, yes. Pinpointing lysosomal iron activation as that crucial first step, that initiation site, that's a major clarification for the field. It's been debated for quite a while.
And Fento-1 itself, it's more than just a research tool, right? It represents a whole new strategy.
It really does. It's a proof of concept for pharmacologically activating lysosomal iron. And it offers a potential way to specifically target those high iron, often drug-resistant cancer cell types like the DTPs.
It provides a kind of blueprint, maybe, for designing other molecules that work this way, exploiting the specific iron metabolism of aggressive cancer cells.
Exactly. It's a new conceptual framework. Instead of just trying to starve cells of iron everywhere, maybe we can target the activation of iron where it's already concentrated and causing problems.
And could this have implications beyond cancer? You mentioned CD44 and iron are involved elsewhere.
Yeah.
Development, immune function.
That's a really interesting thought. The researchers suggest this mechanism, lysosomal iron triggering ferroptosis, might well be relevant in other biological contexts too. Wound healing, immune cell activation, areas where CD44 and iron dynamics are important. It definitely opens up new questions.
So where does the research go from here? What are the next steps this work points towards?
Well, obviously, further optimizing molecules like Fento-1, making them potentially safer or more effective for therapeutic use.
Right.
Exploring those combination therapies is crucial, as we discussed, to tackle potential resistance. And then there's the whole area of exploring the role of lysosomal iron in other diseases, other biological processes. There's a lot to unpack still.
Okay, so wrapping up our deep dive today, this Nature paper really makes a compelling case that activating iron inside lysosomes is a key trigger for ferroptosis, especially in cancer cells.
Yes, it shifts the focus.
And the creation of Fento-1 is a landmark, showing we can actually design molecules to exploit this specific vulnerability, particularly in those aggressive high-iron cancer cells like DTPs. It feels like a genuinely new angle for targeted therapy.
It's definitely a promising new direction. Identifying the lysosome as the starting point and then rationally designing a molecule like Fento-1 to weaponize that location's iron content, that's really elegant science.
It absolutely is. And it really inspires thinking about how we can refine these targeted approaches, maybe combine them, and also look for similar mechanisms elsewhere.
Which leads to the big questions, doesn't it? Thinking about how adaptable cancer cells are. What other specific weaknesses may be tied to other organelles are just waiting to be found and exploited, especially to overcome drug resistance.
And ultimately, the biggest question of all, how do we take these incredible fundamental insights from the lab and translate them into real, effective treatments that actually help patients fighting these diseases?
That's the challenge and the hope driving all this research.